So after a PhD, 5 years, 2 postdocs, 5 years, I've made it. I'm an assistant professor. It's not that glorious. We actually call ourselves Ass Profs. But what am I going to do with 2,000 square feet of lab space, a couple of mostly awesome graduate students, and a couple of mass spectrometers and some startup cash? And of course, I landed, not that you can determine where you land in the academic world, but this is where I landed. Ooh. This, again, for a boy from Southern California that's used to looking at that, I was a fish out of water. All right? But one of the things I dreamed about as a graduate student, as a postdoc, is having a field site close to where I lived, so that I could drive to it in an hour. Yes, it's glorious going to Antarctica and Argentina and New Zealand and Alaska and Africa, and I worked in all those places as a postdoc and a graduate student. But what I cherish now is being able to get in my car and drive to my field site in an hour and take my daughter and my wife and sometimes my dog to the field site. Okay? So it wasn't too bad. So what am I going to do? I'm going to catch animals, of course. That's what I do. I got to learn how to catch the local fauna. Okay? Used to be seals and sea otters, but we don't have those in New Mexico. So now I catch kangaroo rats and grasshopper mice. They're easier to catch. In New Mexico, we have basically 2 families of rodents all across the Southwest. We have these Hetero Mayans, okay? These are food caching granivores. So they have basically, they have very strict dietary preferences. And the cool thing about their ecology is they can cache food. They have like little refrigerators back in their burrows that they throw food into that can sort of supplement their diets in times when things get really bad out there in the real world, okay? And there's a cool body range size here. These guys are so cool. These are the kangaroo rats. They're like softball size, some of them organisms. They hop around on 2 feet like kangaroos across the desert, and they're the funnest things to catch and to study. Okay, so these are almost as cool as sea otters. I know it's hard to believe, but it's true. The other group are the chrysitids. They're the jacks of all trades. Okay, so these are things like pocket mice. Sorry, not pocket mice. Deer mice, grasshopper mice, harvest mice, cactus mice. All right? And these guys are omnivores. They do all sorts of things. In fact, this grasshopper mouse here is the lion of the Chihuahuan Desert. It literally sits up on these mounds in the desert and roars at night. And it eats whatever it wants. Scorpions, millipedes, centipedes, anything. This 20-gram animal will take it all down. Okay? They're nasty little things, and they bite really hard. Okay? So here's the local fauna I decided to start studying. We started trapping them. One of the observations that we could make, because we were working at an LTER site, a long-term ecological research site, is that small mammals increase in abundance when C3 resources are plenty. Okay? So when there's a lot of C3 resources out on the landscape, small mammal populations go up. It appears they like C3 resources. All right, so our goal basically was to build a fairly mechanistic understanding of how climate, precipitation, goes into C3 versus C4 resources, how that impacts resource selection in consumers, focusing mostly on small mammals, and we're going to use stable isotopes and genetic techniques to do this, metabarcoding, how that translates to body condition of animals, and we use a really cool little instrument that I put in a Mercedes Sprinter van, a magnetic resonance instrument that you can take a live animal, throw in there, and it'll tell you the body composition, the fat content, and the lean mass of that animal in 2 minutes or less. And then you take the live animal out and you throw it back in the desert and you catch it next month. It's really cool. And then how does that transition into, or how does that influence survival reproduction? And we did that versus mark recapture, okay? And then population status. So this is our goal for this particular experiment. The cool thing about the Southwest, and you probably know this by now, is that it is a bimodal precipitation regime. I grew up in California. We have a unimodal precipitation regime. That's why there's 40 million people in the state. It only rains between November and March. Well, in the Southwest, most of our rain is actually delivered here in the summer as part of the monsoon. And for reasons that our isotopes have told us back in 1977. This is a young strapping lad that you might recognize. This is some work that he did on cilia in California, looking at quantum yield, which is basically a measure of output, photosynthetic or an energy output for a plant, versus leaf temperature. And there's this interesting crossover temperature that occurs here at about 28 degrees Celsius, where it becomes less advantageous to be a C3 plant versus a C4 plant. Okay? And in New Mexico, it's cool because in the summer, it's greater than 28 degrees C. So a lot of the production that occurs in the summer is C4 production. So the summer monsoon fuels C4 production. In the winter and in the spring, we're below that crossover temperature. So most of the production in the spring months is C3. So it flip-flops back and forth. You don't get this pattern in California. You only get C3 plants in California because the growing season, i.e. when the water comes, is cool enough to only fuel C3 production. C3 plants have an advantage.
So these plants are co-occurring?
Yes.
All throughout the year?
Let's ask the first author.
Excuse me?
These plants co-occur? Same sites?
They have absolutely no overlap in distribution.
Okay.
But those graphics for C3 and C4 are repeated for every single species.
So, Tristan.
So you're talking about production in different areas?
Well, I'm just using this as an example of when C3 versus C4 dominates, when it's basically advantageous.
No, in a place like Albuquerque or a place like Tucson, the C3 and C4 components of the community are there all the time.
Yeah.
But their production rates are highest at different times of the year.
Yeah. And that's actually what I'll show you here, is that for any given year, and here's just 3 years, is that C4 versus C3 production. So this is during the spring, with the blue months, basically, this is precipitation down here, versus the monsoon months. You have basically flip-flopping. You get more C3 production in the spring, you get more C4 production in the monsoon. So that's one of the patterns. So there's a seasonal pattern. But what the point of this is that year to year, there's tons of variability. For example, C3 production in 2005 was pretty good. C3 production in 2006 was awful because there was no rain here in the spring. And then it goes back up when you get a decent spring rains, it goes back up to pretty normal values. So the interannual variability, there's a seasonal variation and then there's an interannual variation. The interannual variation is huge.
Are those values standardized for population sizes or how many of these plants there are?
Yes. This is just above ground net primary production. Yep. So this is just quad drought work basically, and then biomass estimates based on algorithms that compute cover and size of shrubs to biomass in a grams per meter squared basis. Okay, so one thing you can do with this kind of data, we're not going to get into the details, is you can look at species level. What are the different species in the community of small mammals doing in the spring versus the summer? What do they do when C3 production is high versus what do they do when C4 production is high? So that's one thing you can do. So there's pre-monsoon data, there's post-monsoon data. You see species move around in space, in this isotope space over time, okay? If you're moving this way, you're eating more C4. If you're moving this way, you're eating more C3, okay? And again, like I mentioned, if you're in nitrogen space, as you go up this axis, you're eating more insects, and you go down this axis, you're eating more plants, all right? So not surprisingly, all of our chrysalids, our jacks of all trades, have higher nitrogen isotope values than our granivores, because they're more omnivorous. We know that. They eat more insects, but you can see that in the isotope data. That's kind of cool, in the nitrogen isotope data.
Do we have the ability to separate shifts in location versus shifts in the plant signature as well due to pre and post monsoon?
Getting there. Good question. You can also look at population level changes, okay? So this is one particular species. These are pocket mice, tiny little 6-gram things. I don't know why they're called pocket mice because you can fit 10 of them in your pocket, okay? They're tiny, but here you can see pre-monsoon, March through June. This is that springtime. This is how much space and carbon and nitrogen space they're taking up. So basically population level dietary variation. And then that changes in the post-monsoon. They basically expand their dietary niche and they eat different types of stuff. Well, because C4 production is increased and they have a completely different isotopic signature, so that expands the population dietary breadth of this particular species. So you can play these kinds of tricks too. So species level, population level, and then you can look at individual level strategies because we go out every single month and catch these animals. So we end up catching those same individuals month after month after month after month. And this is just an example from 2014. Again, we're looking at pocket mice of individuals that we caught 3 or 4, I think, or more times. Some of these individuals we caught up to 8 times in 1 year, okay? So these individuals are switching all over the place. This is a percent C3 in their diet. So I've taken their isotopic composition and using a mixing model, have transformed that data into percent C3 versus C4 in their diet, similar to what Brian Fry did in 1978, okay? These individuals are generalists. They're switching back and forth between C3 and C4 resources over the course of the year. These individuals down here are C3 specialists. They don't care what's on the landscape. They specialize on C3 resources. So we can get at the proportion of individuals that have these 2 different strategies in the population. All right? This is blood. Good question. So the reason we're collecting blood, as Turi mentioned, is it turns over very quickly, blood plasma. So if we catch plasma. So we take the blood sample, we spin it, we get RBCs, red blood cells, and plasma. The plasma integrates diet over the last 3 weeks prior to capture, more or less. So if we catch an animal monthly, we basically get a monthly resolution of dietary changes. Okay? We couldn't do this with hair, we couldn't do this with red blood cells, we couldn't do this with muscle because the integration window would be too large. So you couldn't look at monthly changes in their isotopic composition. Okay? All right. So we can do all that kind of stuff, but isotopically speaking, what are the assumptions? I've showed you isotope data, different of a tissue. I've showed you estimates of C3 versus C4 diet composition based on their isotope value. So what are some of the assumptions we're dealing with here? So we need to know something about the fractionation or the discrimination between the animal's tissues, in this case plasma, and that of their diet. Okay? So what was the animal eating? We have to know something about trophic discrimination. What's another assumption we're making?
But the things they're eating actually have the same isotopic signature.
Wait, say that again?
Like the things that they're eating have to have distinct isotopic signatures?
Yes.
So just as the gene between the CP.
Exactly. So you have to assume, yes, that you're basically, isotopes don't work if your prey sources all have the same isotope value. So you have to make sure that your prey sources, in this case we know because we're dealing with C3 and C4, which is one of the largest ranges out there, that they have isotopically distinct things. So that kind of gets at my number 3. That's a really good point. Number 2 is exactly what Jeremy said, is that we need to know something about the incorporation kinetics, okay, of plasma, because that's what we're measuring. How much time does that plasma sample represent? All right? And number 3 kind of goes along with your thing, and someone else mentioned it earlier, is that we have to make some assumptions about the isotopic composition of our different food sources and whether they change over time. Okay? As you've learned earlier from Jim's lecture, is that the plant isotopic composition can change in relationship to local conditions. So the way we do that is we constrain that via study design. We actually go out there and measure our plants along with our animals seasonally, just look for changes in their isotopic composition. So we know what those end members are. Okay? So food is a moving target. Okay? Not only does it move around, but isotopically it can move around. So a good study will take that into account and be designed accordingly.